thank you. Hello, everybody. My name is Alexis Martin, and I work as a benthologist in a team of the Muséum involved in the scientific monitoring of the French fisheries in the Southern Ocean. And I'm here to present you a framework I have developed to collect data about Benthic marine invertebrates in order to produce ecoregionalisation and ecological niche modeling studies. I did that in the context of my PhD on the Benthic ecology of the Kerguelen Plateau, and I worked with colleagues who accepted to develop a collaboration, colleagues involved in deep learning and molecular biology. This framework aims at providing solutions regarding the reproducibility of the analysis. An important literature has been produced about the reproducibility crisis in ecology, which is due to the use of massive data and the complexity of the modeling process. But the solutions mainly concern the reproducibility of the computing process and the storage of the datasets, and do not consider the observations of the organisms, which are the fundamental units of the analysis. In that context, a study of Benthic marine invertebrates induces supplementary specific issues such as the lack of experts, the huge amount of taxa which are difficult to identify, the need of abundance distributions to compute models, and the impossibility to conserve all samples collected on field to study them in the lab. To face these issues, we developed a framework which allows to produce comparable results and increase reproducibility in order to understand and interpret the Benthic assemblages, distribution patterns at a large scale, and to build an integrated conservation policy in all the French Southern Ocean territories. This also aims at taking account of the context in the fisheries, of divergent stakeholders, and the necessity to be transparent about data and analysis. The framework is structured into three connected parallel processes. The main process is based on the Bendima database, which is a central point and the main node allowing raw data production and reproducibility improvement. On field, observations of Benthic invertebrates are computerized, including the full photographing of the host organisms. Images are treated and identified in the laboratory to be stored into Bendima. Abundance distributions of Benthic invertebrates are then extracted from Bendima to be used as inputs in the modeling process. The storage of the images within a database constitutes the main key element of reproducibility. Given the possibility of going back to the observation raw data for taxa identification, checking, quality evaluation and corrections, modeling results can be fully assessed, criticized, recalculated or compared. The framework includes also two supplementary parallel processes to improve reproducibility based on the DNA barcoding techniques and the deep learning approach. The first parallel process aims to complete the images of observed organisms with DNA sequences of conserved specimens, and this allows to link the field observations of organisms to real-world data presents about the sampling stations. And the second parallel process aims to record in silico the human process of taxa detection based on the examination of the images of observed organisms. Here you have an illustration of the samples and images collections which is processed on board by scientific observers with bycatch organisms caught with a trawl during a scientific survey in Kerguelen. All the organisms are sorted and photographed, and few samples are conserved in alcohol to be treated in the laboratory. And at the laboratory, all the images are treated by a benthologist, and a crop of each organism is saved into the Bendima database and later identified by a specialist or a benthologist. And you have also the scale, which is extracted in order to extract the size of the organisms. Here is an illustration of the DNA barcoding part of the project, with pictures showing the sorting of the samples, tissue extraction, and the DNA extractions. And you have the information about the markers we used for this part of the projects. The use of the framework allowed us to build a database including observation of more than 90,000 of organisms since 2015, and to date, considering the state of the identification work, they fall into 110 taxa with organisms identified from the species to the phylum level, and half of the database is identified at the species level, I hope to reach maybe 130 taxa when I will have finished this part of the job. And you have on the slide the spatial distribution of the data. And 13 phyla are represented, including an important data set for cnidarians, sponges, echinoderms, and ascidians, and many abundant distributions we can produce on those taxa to compute models. The deep learning part of the project started 2 years ago when Benthic reached a sufficient level regarding the amount of data and the precision of the identification work. An algorithm has been trained on the contents of Benthic in order to obtain a network which is able to detect the organisms in the pictures and to classify them, to identify and attribute a taxon to the organisms. I'm sorry, there is a little mistake on the slide. Actually, the network is not a fully connected neural network for those who are involved in such analysis. It's a convolutional network using 2 parts in the analysis. And actually, the fully connected neural network is one which is able to identify the pictures and to classify the observations. And there is another part in the network, the faster CNN algorithm, which is able to detect organisms in the picture. And the network, the algorithm has been trained on the contents of Benthic, and the network which reproduces a human identification process, or the bentologists who identify the contents of Benthic can be stored itself in a database and shared. This is what is represented bottom of the picture. And when we have trained the neural network and the artificial intelligence is able to detect the organism and to identify them, you can then give it new pictures, and then the artificial intelligence is able to identify the organism on the pictures and to give an identification. So on the top you have, on the left side you have what we have done with human brain, and on the right you have an example of the application. The computer have classified and identified the organisms. Here you have an example of the use of the artificial intelligence, which is able to detect all the organisms and to identify them at the level of large groups. And this result with sea stars is very interesting because the network is able to detect the organisms even they are photographed with an important overlap. You can see that all the squares are okay, and the network could identify all the organisms. And here you have an example with ophiurids and a piece of coral, which are also well detected and identified by the network. And on these pictures, there's no problems of overlapping between the organisms. And this is a picture with ascidians, and also well perfectly detected and identified by the network. Even the picture is very complex and hard to check. And the last example, you have here a very interesting and impressive result for me. In this picture, the network could perfectly detect and identify all the crinoids with a complex form and a very important overlap between the organisms. And there is a little mistake in the picture, but for me it's the most impressive result. The artificial intelligence could not, we haven't shown it seaweed pictures, and so when he see seaweed, he thought that it was corals, and this is very impressive. And you have also a gastropod on the left, which have been in the middle of those organisms well detected. I will finish the presentation by providing a concrete example of the use of the framework when we need to assess and compare modeling results. We have computed an ecological niche model for a crinoid, the one which was on the former picture, Promacrocrinus kerguelensis, by extracting the reliable abundance data from Benthic. Moreover, we replaced 20% of the data with another crinoid from the same family, Phlorometra mosoni, in order to simulate the effect on the modeling results of an error of identification based on the confusion between two similar-looking species, affecting 20% of the observations. And we also replaced 20% of the data with observations of organisms of the same family, but which could not be identified at the species level. And this aims to simulate the effect on modeling results of an error of identification based on overstatement and affecting also 20% of the observations. Here you have the predicted distribution pattern we obtained with the model computed with the reliable data. You have the predicted abundances, and in yellow the localization of the protected areas in the northern part of the Kerguelen plateau. And it's possible to calculate the protection level for the species by comparing the sum of the predicted abundances located within protected areas to the total of the predicted abundances. Here you have the comparison with the two models computed with datasets including 20% of misidentifications. And you can see that the distribution patterns are different. So misidentification also impact the expected abundances and the protection level that you have. So this is the predicted abundance by the model, and this is all the abundances located in protected areas, and this is the protection level you can calculate with those values. And so you can see that the conclusions concerning the distribution pattern, but also the protection of the species are very different if we have 20% of misidentification, and it's different according to the case of misidentification. But if the modeling process is only based on a dataset with observations in the form of table of abundances, which is common in ecology, and we have no access to the organisms' observations, how can we assess the results and compare the models? On the left you have the model have been computed 100 times, and here you have the standard deviation map of the reliable model, but only regarding the calculation process, not the observations. And to obtain maps of uncertainties, regarding the observations and to assess the models, those two maps, we need access to the observations. With Benthic, we can evaluate a model regarding both the computing process and the observations, which are the fundamental units of the analysis. In this example, you can assess the raw data and perform corrections on the identifications of the organisms. You have access to the observations, you have also access to the network of the identification, and you have access to the samples with molecular data. And this allows to produce a new model based on corrected data. And it's impossible to obtain maps of uncertainties based on the identifications by comparing the various abundances. And you can see that here you have uncertainties in a part of the distribution pattern. This example is about the issues regarding misidentifications, but it's just an illustration. The same concept and same process can be undertaken if we need to compare models from two different areas, or models in the same area, but in different periods. So this is one of the examples of the use of the framework to discuss the models. And thank you for your attention. I would be happy to answer you if you have questions. And those last pictures is to show you how Benthic's pictures can be confusing.